Good morning. My name is Haley Wang, and today I'll be presenting our work on how childhood maltreatment relates to deviations from normative brain structure using data from over 3,000 individuals from the ENIGMA PTSD and MDD working groups. I'd like to thank the organizers for the opportunity to speak in this exciting symposium on mapping predictors of disorders. And I have no disclosures. We know that childhood maltreatment, including abuse and neglect, is incredibly common. It affects over 50% of the general population, and it's also one of the most significant risk factors for developing stress-related disorders like major depressive disorders and PTSD. Two major types of childhood maltreatment are abuse and neglect. Abuse could be understood as the presence of harmful input, whereas neglect is characterized by the absence of positive input. And moreover, research has found that they might also create distinct subphenotypes of patients with MDD and PTSD, indexing earlier age of illness onset, more severe symptoms, and lower chance of response to therapy or medication. And the central unanswered question is how these early experiences translate into later vulnerability. A leading hypothesis is that maltreatment disrupts normative brain development, but the exact nature of these changes has been unclear. Previous research has provided a crucial foundation, consistently pointing to structural alterations in brain regions critical for threat processing and emotion regulation, like the hippocampus and prefrontal cortex. And for example, here is some of the abuse type specific effects on the developing brain from a review paper in 2016. However, the findings have been notably inconsistent, and this variability likely stems from an array of issues, for example, challenges like smaller sample sizes and the tendency to use group diverse experience of abuse and neglect together, which may obscure more specific effects. Past research has given us important clues, but has also faced some additional key limitations. First, the brain develops dynamically, so the impact of stress may depend on developmental timing. Second, we see clear sex differences in both brain maturation and disorder prevalence, but many studies haven't had the power to examine these effects. And finally, lumping different types of maltreatment together, like abuse and neglect, may obscure distinct biological pathways. And to move forward, we need an approach that can precisely quantify how an individual's brain deviates from a healthy developmental trajectory. We need a very large data set to tease apart these nuanced effects. And to do this, we leveraged the power of the ENIGMA consortium, analyzing data from over 3,700 participants across 25 sites. We assessed childhood abuse and neglect using CTQ and processed structural MRI scans with Reservoir. The core of our approach is normative modeling, using the Centel Brain tool, which has been trained on over massive amount of healthy individuals. We calculated how much each person's brain structure deviates from what we could expect from their precise age and sex. For each phenotype as well. This gives us a deviation score for every brain region, which we can then correlate with their trauma history. So what did we find? We stratified by developmental stage, children, young adults, and older adults, and analyzed males and females separately. A key discovery is that these associations were not static across the lifespan. The most widespread and robust effects were observed in young adults and with generally weaker patterns in older adult cohort. While we didn't find any significant correlations in our pediatric cohort, this requires a nuanced interpretation, as our power analysis confirmed that the pediatric group was underpowered to detect some of the most subtle effects we saw in adults. However, that same analysis also showed that most robust findings from the adult cohorts, specifically later I'll show you in frontal lobe surface area, should have still been detectable. And since these effects were absent in the pediatric group, it suggests that the lack of association in childhood may not be solely due to limited power and could potentially point to true developmental differences. Focusing first on subcortical volumes, we saw distinct patterns across adulthood. In young adults, abuse was linked to larger thalamus and pallidum volumes in both sexes, but only females showed smaller hippocampus and putamen volumes with both abuse and neglect. In the older adult cohort, this picture shifted slightly. Some effects persisted, like the larger pallidum, but new patterns also emerged, such as smaller amygdala volumes in males exposed to abuse. And this tells us subcortical signature of maltreatment is not so static. And for cortical thickness, we observed a strong differential pattern between age and sex. In young adults, abuse was linked to thinner cortex in several higher order regions, contrasted with the thicker medial or frontal cortex. And interestingly, this pattern of widespread thinner cortex persisted into older adulthood for females. And for males, however, these associations were no longer significant in the older cohort. This suggests that a potential sex difference in the long-term persistence of these cortical thickness alterations. The patterns for cortical surface area were particularly interesting. The strongest effects were in young adult females who showed a complex reorganizations, a smaller surface area in parietal networks and larger area in frontal regions, linked to both abuse and neglect. And what's remarkable is that for abuse, this widespread pattern of surface area alteration persisted into adulthood, older adulthood in females, suggesting that it may be a highly enduring neural signature. And in contrast, the effects of neglect appear to be confined to the young adult window. And to ensure that our results were reliable, we ran several sensitivity analyses. Crucially, we found that the associations between maltreatment and brain structure persisted even after accounting for a psychiatric diagnosis. And furthermore, the results were not driven by any single data set or site. This gives us the confidence that we are observing robust transdiagnostic effects of childhood maltreatment. And due to the lack of control population in some sites and other covariates that differ by sites, we could not remove these potentially clinical meaningful effects. So in the main results we're presenting here, we're doing the version without applying site harmonization. However, to be cautious, we did perform it as a supplementary analysis, and harmonization was come back and left the main pattern intact. And to summarize our key results, the neurobiological deviation patterns of childhood maltreatment is both specific and dynamic. This effect is varied by age, with some patterns persisting while others change in older adulthood. They are sex-specific, with females often showing more enduring alterations, and they're trauma-specific, with abuse having more lasting effects than neglect. And finally, they're concentrated in higher order association and limbic surface. So what does this all mean? The concentration of effects in young adulthood, especially in late developing association cortices, suggests that this is a critical time for the manifestation of these structural changes. The persistence of many of these patterns into later life, particularly in females, underscores their enduring nature. And clinically, this underlying neurobiological diversity could be one reason why patients with trauma histories often respond so variably to our standard treatments. And a critical limitation, especially relevant for this symposium, is that our study is cross-sectional. We are observing age-related differences, not developmental changes within individuals. And our findings provide a rationale for future longitudinal studies, which are essential to directly map these developmental trajectories. And in conclusion, our work here in this large ENIGMA sample demonstrates that childhood maltreatment leaves a lasting but not static mark on brain structure. And these deviations are specific to age, sex, and trauma type, and they follow a dynamic course across the lifespan. And by mapping these specific neurobiological profiles, we move one step closer to understanding the mechanism of risk and ultimately to developing more tailored and effective interventions for those who have experienced early adversity. And I'd like to extend a huge thank you to the ENIGMA, MDD, and PTSD working groups, all the participating sites, our funding agencies, and most importantly, the thousands of individuals who contributed their data to this research. This work was done with the support of Dr. Tiffany Ho and the UCLA Candy Lab. The manuscript preprint is at this QR link, and thank you everyone for your attention and time.
So feel free to come up to the mic in the center of the room if you have any questions.
Yes.
Going once, going twice. All right, yes.
Actually, I'm also using the ENIGMA data. I wonder that how do you deal with different measurement of childhood adversity? Because some of the study sites use the CT2 questionnaire, but some of them they use the ACE or something else. How do you, yeah.
Right, so that's a great question. Harmonization of data is always an issue when you're working with multi-site data and trying to collate it together in a post-hoc manner. So we actually only included sites that had the CTQ. So if they had the ACEs, they were not included in this particular analysis. But the CTQ appears to be one of the most commonly used measures of childhood maltreatment. So as you can see, we were able to get quite a large sample size.
Yeah, it is. And the second question is that I wonder if in your model you control if the effect of socioeconomic status.
Right, so socioeconomic status was not consistently measured in the same way across all sites. So if you check out the preprint that Haley has posted, we attempted to do a sensitivity analysis for sites that had information such as highest parental education. Income data was a lot trickier to work with, but that would be, for a subset of sites, we were able to look at that and to see that our results pretty much remain the same when adjusting for parental education.
Thank you.
Yeah.
I'll just ask a question because your title is normative brain data patterns and so on. But I'm expecting that you have normative trajectories among the people, then you compare the CTQs, the patients with the normative data. So is that what's happening in the analysis, or is just group comparisons in
Right,
such a way?
so these are cross-sectional data, and the normative modeling approach allowed us to compute for each person in a given region the deviation score from what would be typical for that person's age based on the centile brain model. So these are not longitudinal data. We were not looking at trajectories or deviations from trajectories per se.
So in other words, you did compute the deviations of individual from the normative data.
Correct.
Then you basically compare, you're basically looking at those measures and see how that's happening in the young people with trauma. And
correct. So for each age and sex-based group, we were looking at the association between CTQ or childhood maltreatment scores with these deviation scores for each region of interest.
Thank you.
Yeah.